We've already learned that cellular respiration is the oxidation of organic compounds to release energy, which is captured as ATP. In this video, we'll learn the specific chemical reactions of glucose oxidation, the steps at which energy is captured, and the cellular structure where all this happens, namely the mitochondrion, and that's where we'll start. The mitochondrion is a double membrane organelle. Here you see the outer membrane, which is in contact with the cytoplasm, and the highly convoluted inner membrane. All of these convolutions create multiple surfaces called cristae. This architecture divides the mitochondrion into three functional compartments. First, there is the space between the inner and outer membrane, called the intermembrane space. There is the space inside the inner mitochondrial membrane, called the matrix. Finally, there are the cristae themselves, or the surface of the inner mitochondrial membrane. Note that these many convolutions give the inner membrane a very large surface area, and, as we'll see in a bit, this large surface area is where ATP is made. So the larger the surface area, the more efficiently the mitochondrion can make ATP. So the structure of the mitochondrion allows it to carry out its function. To help us understand cellular respiration, it is helpful to think of it in three stages: glycolysis, pyruvate oxidation, and citric acid cycle, and oxidative phosphorylation. We will look in detail at each of these stages, but for now, let's just get an overview. The first stage of cellular respiration is glycolysis, and it takes place in the cytoplasm. The oval at the bottom represents a mitochondrion, so everything else on the page is the rest of the cell. Or what's outside the mitochondrion is the cytoplasm, and that's where glycolysis happens. Let's think about that word for a moment. Glycolysis is glyco, which means sugar, and lysis, which means breakdown. So glycolysis is the breakdown of sugar, and that's exactly what happens. Glucose, a 6-carbon sugar, is broken down into two 3-carbon molecules of pyruvate. Pyruvate, which is generated in the cytoplasm, moves into the mitochondrion where the rest of cellular respiration happens. So pyruvate is an important transition molecule. The next stage in cellular respiration is pyruvate oxidation and citric acid cycle. These both happen in the mitochondrial matrix. Note that it is in the second stage that carbon dioxide is released. Remember the net equation for cellular respiration. All the carbons of glucose are eventually released as carbon dioxide. That all happens in stage 2. The third stage of cellular respiration is oxidative phosphorylation. That's a mouthful, so we'll use the short form OXPHOS. OXPHOS is the combined process of electron transport and chemiosmosis. Let's start with electron transport. Recall that the product of glycolysis, pyruvate, feeds the next step in cellular respiration, pyruvate oxidation. The product of pyruvate oxidation, acetyl-CoA, feeds the citric acid cycle. The product of pyruvate oxidation and citric acid cycle are high energy electrons carried by NADH, and it is NADH that feeds OXPHOS. These electrons, that's the electron transport part, where those electrons travel to oxygen to form water. Chemiosmosis is how the energy of those electrons is used to make ATP. Speaking of ATP production, let's take a look at the relative contribution of each stage of cellular respiration to total ATP production. We'll see this in more detail in just a bit, but for now, glycolysis generates 2 ATP, as does the citric acid cycle. But the bulk of ATP production comes from OXPHOS, 28 ATP. That makes a total of 32 ATP, but that's just an estimated average. It varies from species to species and under different conditions, but we'll use 32 as a solid estimate. Remember that cellular respiration is about making ATP, which in turn powers all the cell's operations. When I say make ATP, I mean something very specific: the phosphorylation of ADP to ATP. This of course requires energy, and that energy, as we're learning, comes from the oxidation of organic compounds. Making ATP happens in two ways: substrate level phosphorylation and oxidative phosphorylation. When we cover photosynthesis, we'll learn a third way, photophosphorylation. In substrate level phosphorylation, a phosphate group is transferred from an organic compound to ADP to form ATP. This is essentially reaction coupling again, but in this case, phosphate is transferred to ADP from a very high energy molecule. ADP phosphorylation is an endergonic reaction, but the energy required is exceeded by the energy released when phosphate is hydrolyzed from another organic compound, and the reaction is exergonic overall. This is what happens during glycolysis and the citric acid cycle, and does not require oxygen. Oxidative phosphorylation uses the energy release by the flow of electrons from organic compounds to oxygen to make ATP from ADP and free phosphate groups. Remember from the last slide that most ATP production is from oxphos, so ATP production with oxygen is much more efficient than without oxygen. Okay, let's get into the specifics of cellular respiration, and we'll start with glycolysis. Don't be alarmed. This is a very complicated slide because it shows all the structures of the intermediates of glycolysis. You don't need to know any of the structures. I use this figure anyway because it illustrates several points rather nicely. Let's start at the beginning with glucose. Glycolysis is a 10-step process that converts glucose, a 6-carbon molecule, into 2 molecules of pyruvate, a 3-carbon molecule. Glycolysis can be divided into 2 phases, an energy investment phase and an energy harvesting phase. In the first phase, energy is invested in the form of ATP at 2 different steps to phosphorylate a sugar molecule. ATP is used at the first step and again at the third step. The net result is a 6-carbon sugar that is doubly phosphorylated, or more properly, phosphorylated at 2 different sites, due to an investment of 2 ATP molecules. That 6-carbon sugar is then broken down into 2 3-carbon sugars. The rest of the pathway shows what happens to one of those 3-carbon molecules, so keep in mind that these steps are happening twice. Having invested 2 ATP, the second half of glycolysis harvests energy in the form of ATP. There are 2 steps from this harvesting phase that generate ATP via substrate level phosphorylation, here at steps 7 and 10. Since these steps happen twice for every glucose oxidized, that's a total of 4 ATP molecules harvested. 4 molecules of ATP harvested minus 2 molecules of ATP invested leads to a net yield of 2 molecules of ATP. The last thing to point out is that step 6 also generates a molecule of NADH, the electron-carrying intermediate that we already mentioned. It happens twice per glucose molecule for a total generation of 2 NADH. NADH will carry electrons to the mitochondrion where their energy will power the generation of ATP by oxidative phosphorylation. If that slide was too much for you, here is the abbreviated version, and it has all the information you need to know. Glycolysis is the splitting of glucose into 2 molecules of pyruvate with a net production of 2 ATP and 2 NADH. It happens in 2 stages, an energy investing phase where ATP is used to phosphorylate sugars, and an energy harvesting phase where phosphate groups are transferred to ADP to make ATP. The net yield is 2 ATP and 2 NADH. It is important to note that all the reactions of glycolysis happen in the cytoplasm. We haven't entered the mitochondrion yet. The next stage of cellular respiration is pyruvate oxidation. The diagram depicts 3 steps in pyruvate oxidation, and we'll go over them. But before any of that happens, pyruvate, the product of glycolysis, moves into the mitochondrion. So step 0 is translocation to the mitochondrion. Pyruvate is first decarboxylated. That is, this COO negative is lost as carbon dioxide. Note that this is the first step that releases carbon dioxide. Eventually, all 6 of the carbons of glucose are released as carbon dioxide. After decarboxylation comes oxidation. As pyruvate is oxidized, NAD+ is reduced to NADH. Remember, oxidation and reduction go hand in hand. Finally, this CoA molecule is added to make acetyl-CoA. What is CoA? Coenzyme A, or the coenzyme of acetylation, is a small organic molecule made from vitamin B5 or pantothenic acid. It is essentially a 2-carbon carrier. Acetyl is a 2-carbon unit, and CoA is going to carry it from pyruvate oxidation to the next step, the citric acid cycle. In summary, pyruvate oxidation happens in the mitochondrion and is the conversion of pyruvate to acetyl-CoA with the loss of one carbon as carbon dioxide and the generation of one NADH. Since 2 pyruvate are generated per starting glucose, you should think of these reactions as happening twice. Once again, there's no need to be alarmed. This is the citric acid cycle with the structures of all the intermediates. You don't need to know the structures of the intermediates. I use the illustration only because it makes several important points. The citric acid cycle is also called the TCA cycle, the name that I grew up with, and the Krebs cycle, after Hans Krebs, the man who first described it. Let's start at the top and you'll see acetyl-CoA, the product of the previous step, pyruvate oxidation. Remember that CoA carries 2 carbon molecules, acetyl. So CoA is taking these 2 carbon molecules and dropping them off in the citric acid cycle. Then it will go back and get 2 more carbons, which will also add to the cycle. The 2 carbons of acetyl-CoA are added to a 4-carbon molecule. Its name isn't important, to form a 6-carbon molecule, citrate, or citric acid, which gives the cycle its name. Note also that citric acid has 1, 2, 3 carboxylic acid groups, giving the cycle its other name, the tricarboxylic acid or TCA cycle. Again, the names and the structures of the intermediates aren't important. Here is what is important. 1. It's a cycle. 2 carbons enter and 2 carbons leave. All the intermediates are regenerated. 2. Carbon, 2 of them, enter as acetyl-CoA and 2 carbons leave as carbon dioxide at steps 3 and 4. Note that by now we've eliminated all the carbons of glucose. We lose 2 carbons as carbon dioxide for each acetyl-CoA that enters the citric acid cycle. But remember that each glucose generates 2 acetyl-CoA, so we've effectively eliminated 4 carbons through the citric acid cycle. And we eliminated 2 carbons at the previous step, pyruvate oxidation. That means we've accounted for all 6 carbons of glucose. 3. 1 molecule of ATP is generated at step 5. This is another example of substrate level phosphorylation. I know, it says GTP, which is a different molecule than ATP for sure, but energetically they're equivalent. Your textbook doesn't even distinguish between them. Remember that each glucose yields 2 acetyl-CoA, so there are 2 ATP per glucose generated at this step. 4. 3 NADH are generated, steps 3, 4, and 8, and 1 related molecule, FADH2. Like NADH, FADH2 is a 2-carbon carrier, and that's all you need to know for now. That's 6 NADH and 2 FADH2 per glucose. 5. We've dispensed with all the carbon of glucose, but we still have the high energy molecules in the form of NADH and FADH2. We harvest energy of those electrons in the final stage of cellular respiration, oxidative phosphorylation. Before we go over that, a quick summary. To summarize, the citric acid cycle uses 2 acetyl-CoA per glucose and generates 4 carbon dioxide, and now we've accounted for all 6 carbons of glucose, 2 ATP, 6 NADH, and 2 FADH2. On to the last stage of cellular respiration, oxidative phosphorylation, or OXPHOS. I hope you didn't find the previous sections too confusing, because things really get complicated from here. Now that we've dispensed with all the carbon from glucose, all we have to deal with are the high energy electrons that NADH and FADH2 carry. OXPHOS, which is the combined processes of electron transport and chemiosmosis, is how the mitochondrion uses the energy of those electrons to generate ATP. This is also the step that uses oxygen and generates the most ATP. Here is a quick summary. In electron transport, the electrons in NADH and FADH2 flow to oxygen and give up energy as they do so. That energy is used to pump protons out of the matrix to generate a proton gradient. The energy of the proton gradient, in turn, is captured as ATP when protons flow back across the inner mitochondrial membrane through ATP synthase. Let's look in more detail at each of these processes individually, starting with electron transport. Before we get into the substage of electron transport, let's review the structure-function relationship of the mitochondrion. The important components of oxphas are arrayed along the inner mitochondrial membrane in a precise order. As such, they're part of the barrier between the mitochondrial matrix and the intermembrane space. Because of their position along the inner mitochondrial membrane, they are poised to interact with NADH and FADH2 generated by the citric acid cycle and pyruvate oxidation in the mitochondrial matrix. Remember that the inner mitochondrial membrane has a very large surface area owing to its many convolutions. So electron transport is only possible because of the precise and complex structure of the mitochondrion. This is the electron transport chain. Electrons enter the chain when NADH or FADH2 give them up, or when NADH or FADH2 get oxidized. The electrons are carried by various intermediates, numbered 1, 2, 3, and 4 here, but that's not important. What is important is that the electrons eventually travel to oxygen, which accepts the electrons and protons to form water. The electrons are giving up energy as they travel to oxygen. That energy pumps protons out of the matrix and into the intermembrane space, establishing an electrochemical gradient across the inner mitochondrial membrane, or you could call it a proton gradient. Proton concentration is high in the intermembrane space and low in the matrix. This proton gradient is a form of stored energy. That energy will power the production of ATP in the next step, chemiosmosis. Chemiosmosis means using the energy of the proton gradient to drive ATP synthesis. This is accomplished by AT synthase, an enzyme that sits in the inner mitochondrial membrane. Remember that, due to the electron transport chain, there is a proton gradient across the inner mitochondrial membrane. Proton concentration is high in the intermembrane space and low in the matrix. Protons that flow down this concentration gradient are giving up energy. That energy is used to basically turn a gear in the ATP synthase that forces ADP and phosphate together and generates ATP. Note that in this case, the phosphate is free in solution, not attached to an organic molecule or anything else. That's what PI means, inorganic phosphate. So ATP production is powered by the energy of protons flowing down their concentration gradient through ATP synthase. This is analogous to water running downhill and turning a turbine that then generates electricity. To summarize, oxphas is the combined processes of electron transport and chemiosmosis. Electron transport uses the energy of electrons in NADH and FADH2 to generate a proton gradient, as those electrons flow to oxygen to form water. The energy of the proton gradient, in turn, is captured as ATP when protons flow down their concentration gradient across the inner mitochondrial membrane through ATP synthase. Putting all these stages together, glycolysis generates 2 ATP by substrate level phosphorylation plus 2 molecules of NADH, which contains high energy electrons to be used later. Pyruvate oxidation doesn't produce any ATP directly, but does produce 2 NADH. The citric acid cycle produces 2 ATP by substrate level phosphorylation plus 6 NADH and 2 FADH2. These 10 NADH and 2 FADH2 contain high energy electrons that are used in oxphas to make 28 ATP. Cellular respiration is about oxidizing organic compounds and capturing the energy released as ATP. In our next video, we'll see alternate ways to make ATP from organic compounds. After that, it's photosynthesis, which is how these organic compounds are made in the first place.